Thank you so much for joining us virtually this evening. Since 1931, Berkeley Lab has been dedicated to advancing the scope of human knowledge and seeking science solutions to some of the greatest problems facing humankind. Tonight, you'll hear about some of Berkeley Lab's contributions to the fight against COVID-19. Before we begin, let me briefly explain how we'll manage the presentation. You'll notice that your video and audio were automatically turned off when you join the meeting, but you'll be able to see the speakers and their presentations once we start. We're recording the webinar so that people who are unable to watch it live can access it later. And I want to let you know that there will be a Q&A session after the presentations, and you can submit questions anytime during the presentations using the chat function. In your controls at the bottom window, simply click on the chat icon, type your question, and press enter to send it. We hope to host additional virtual presentations dedicated to highlighting the research at Berkeley Lab. If there are any topics you're interested in hearing about at future events, please send us your suggestions using the chat function. Before I introduce our first speaker, I'd like to say thanks to my colleagues in Government and Community Relations, Strategic Communications, and IT. This event would not have happened without their help. Thank you. With that, it is my pleasure to introduce our first speaker, Rebecca Abergel from the Chemical Sciences Division.
Well, thank you for dialing in. My name is Rebecca Abergel, and I am a faculty scientist in the Chemical Sciences Division, and I will be telling you a very brief story about how we've pivoted some of our science to try addressing pressing issues in responding to the COVID-19 pandemic in the past few months. And so first I need to introduce what we do a little bit. And so I lead the heavy element chemistry group, which is in the Chemical Sciences Division. And this is a fairly well-established program of the Department of Energy, where we are focusing on looking at the fundamental properties of the heavier elements of the periodic table that you may not necessarily hear about. And you may know our program for legacy work that happened quite a while ago, for example, the discovery of Berkeley. But mostly we are very skilled in working with a lot of different hazards, including radioactive elements and biological hazards. And we have access to a lot of state-of-the-art instrumentation that is really helpful in developing new studies for looking at materials in very small amounts or materials that may be fairly dangerous to work with. In addition to looking at the fundamental properties of those heavier elements, we've also spent the last 10 years preparing for what could be a nuclear emergency. So this is not a viral pandemic, but there are a lot of similarities between trying to getting ready to respond to a nuclear emergency in comparison to reacting to a global pandemic. And so what we've done in the past 10 years is participating in large programs that are aiming at developing responses and preparing the population so that we have increased resilience were there to be an emergency, a nuclear accidents, or exposure to the population. We've also been developing new medical countermeasures to be included in the United States stockpile. And by medical countermeasures, I mean pharmaceutical that will be stockpiled by the government so that they could be distributed in a very rapid manner to the population to help responding to, for example, the contamination event with those heavier elements. And this work in particular involves coordinating not only the technical aspects of pharmaceutical development, but anything for planning the manufacturing of these pharmaceuticals, the distribution of these pharmaceuticals, and coordinating the response through different federal agencies throughout the country. And that involves interacting on a very regular basis with the Food and Drug Administration, the FDA, or the Center for Disease Control, the CDC. And so if you think about this and developing either new pharmaceuticals as therapeutics or new diagnostic platforms to respond to an emergency and help the population, this is where the similarity kicks in between preparing for a nuclear emergency or a pandemic, a biological hazard. And so this is what we have been doing in the past 2 months in pivoting all of our expertise in coordinating different agencies and developing new platforms for diagnostics and therapeutics by leveraging all of our infrastructure, instrumentation, and workforce. And so we've been participating in a national lab-wide response to coordinate some research and deployment strategies in relation to testing the population for COVID-19 infection. And so, again, this is really a very large effort that involves a lot of people in different national laboratories, including ours, Berkeley Lab. It involves working with the FDA, the CDC, and other federal agencies. So not only the Department of Energy, but the National Institute of Health or the Department of Defense. And the main goal of the portion of this project that we've been leading at Berkeley Lab is to develop and establish some alternatives in terms of instrumentations and reagents so that we can provide those for new national guidelines and increase the efficiency of testing in the United States. And so I will very briefly tell you about what this really entails. So as probably some of you know, the current testing is based on a type of assay that is called RT-PCR, that stands for reverse transcriptase polymerase chain reaction. And this is a method that is used to detect and quantify RNA in a specific sample. And so this is a very well-developed method for evaluating whether somebody has been infected with the virus. However, there is a lot of steps and hurdles to go through when you are developing such a diagnostic test. And what the specific test for COVID-19 entails is going through different steps. One of which is collecting different specimens, so interacting with patients and trying to collect whatever we will be using to determine whether there is viral presence in the specimen, and that could be fluids from so body fluids, including saliva, or the fluids that you're collecting by swabbing the nose or mouth. The next step would be transporting the specimen and making them available for analysis. Then you would be extracting the RNA from those samples, and so that entails a series of steps in terms of making sure we have valid samples to undergo the next phase of testing that is then the PCR assay, in which we amplify DNA that is templated after the RNA that has been extracted from our specimen. And then further on, if we're not wanting to be constrained by the number of tests that we can perform, we want to develop some automated process so we can have a high throughput workflow in order to make sure that the testing is efficiently performed in the country. And so across all of those different steps, we have identified many aspects in which we can contribute and that we can improve. For example, if you think about the specimen collection for COVID-19, you have probably heard about those nasopharyngeal swabs, or NP swabs, which is the prevalent method to collect a specimen. And those are not very, they're not simple, they're not fast, they're not easy. And so we've been testing various methods to collect, for example, saliva samples, and trying to assess whether saliva samples would be as viable in terms of specimens as those NP swabs. The next phase, which would be transporting our samples in some viral transport media or some specific fluid, needs also to be assessed, in that you want to make sure that this process is sustainable regardless of the temperature, the time the specimen is being stored or transported all the way to a facility where you can do the assay. And how, if you are freezing the samples, how are they frozen, how fast are they thawed. So all of this temperature dependence and time dependence studies are necessary to understand how these samples are being are kept viable all the way from collection to analysis. The next step, which involves the RNA extraction, is very costly, or involves a lot of costly reagents for which there has been a shortage at some time points in the last 3 months. And so we have also been focusing on developing new reagents and new processes for either improving the RNA extraction step or completely bypassing it. And we've also worked on evaluating and developing new reagents for streamlining the amplification process through the RT-PCR assay. And finally, something that is very unique to Berkeley Lab is developing a high throughput platform so that we can increase the number of tests that can be done in a shorter amount of time, using all of the automated instrumentation that we have access to at the Joint Genome Institute on site. So this is really just a very brief overview of this effort that has been coordinated to improve testing as a platform to develop new national guidelines and help the CDC and the FDA issue those new national guidelines. So that is pertinent, for example, for to workers who are returning to work, to devising new policies and making sure we have testing available so that we can inform the population. And overall, this effort has been coordinated between many national labs, including the Los Alamos National Laboratory, Sandia National Lab, Pacific Northwest National Lab, Lawrence Livermore, Oak Ridge, and Argonne National Labs, including Berkeley Lab as well. And so you can imagine that it involves a lot of infrastructure and a lot of coordination between teams. And on the Berkeley Lab front, it has been a collaborative effort between two different areas, the biosciences and the energy sciences. And it has involved people from different divisions within those areas, but specifically from the Joint Genome Institute and from the ALS as well. And so with this very short introduction to our science pivot towards developing new testing reagents or alternative reagents and instrumentation for testing, I'm going to end it here and introduce our next speaker, who is Michael Connolly from the Molecular Foundry.
A quick interruption. This is Jen from the Government Relations Office. We were just joined by our lab director, Mike Witherell, and we're going to turn it over to Mike right now so he can begin his remarks. Thanks, Rebecca.
So, and thank you, Rebecca, and thanks, Jen. Thanks for joining us this evening. I want to start by my remarks by saying something that's not about the COVID research, but that our community is suffering from unimaginable grief that caused by repeated events of race-related violence we've been going through in this country. So this has been a very important discussion in our laboratory. Every institution in our country, including our laboratory, needs to discuss more directly the impact of discrimination and prejudice, conscious and unconscious. And that's something I and the lab's leaders are committed to making our laboratory more inclusive and welcoming to our underrepresented minorities. So that's something that is something very important to me that we're going to take on in the days and weeks to come. So For the past 3 months, all of us have been at the laboratory, have been figuring out how to work and live in a new environment caused by this pandemic. The health and safety of our staff and of our community are our top priorities. So I'm in the situation where I have the personal responsibility to protect the health of everyone who works at the laboratory. So, of course, when the shelter in place went into effect, we removed most of the people from the laboratory site to be working from home. And a very small group of people have been working in our facilities, which enabled us to conduct the critical COVID-19 research that you're hearing about this evening. Most of the staff have been teleworking from home and have shown remarkable resilience in carrying out world-class research in this environment. In fact, it's been amazingly productive for many of the people who work at the laboratory over this time. Of course, much of what we do needs the facilities of the laboratory, however, and we are on our path to working back towards using those. We've established a strong safety culture among the people who are working at the laboratory based on physical distance, face covering, hand washing, constant cleansing, and special training, the things that everyone's becoming familiar with. And we've based our practices during the pandemic on federal, local, and CDC guidelines. Just this week, on Monday, we started the process of gradually increasing the number of people on our main site and off-site locations. The goal is to slightly expand our mission essential research while continuing this COVID-19 research you're hearing about this evening. So about 18% of our staff is now at one of the lab sites on a typical weekday now. We have a lot of practices in place to make sure that everyone who does that is safe, wearing masks, maintaining social distances, and so forth. And we have everyone at the lab also takes a health pledge committing to follow the requirements and best practices to make sure that we all are working by these rules. Still, most of our staff are teleworking and will continue to telework in the next few months, probably for the rest of the year. And that's how we are facing this new environment that we're operating in. So I'm going to now show you a couple of slides just to further talk about what we're doing. As we said, we have taken a sudden correction of our direction for research laboratories. So now a significant part of what we're doing is working on many aspects of COVID-19 research. You've heard already about our work on testing. Let me go back and put this in some larger perspective. The fact is the national laboratories were invented and established to provide science and technology to the country. Scientific solutions addressing national priorities, energy security, resilience, economy, the environment, and health is becoming an increasingly important part of this. We do discovery science of the type that, for example, Saul Perlmutter did in his Nobel Prize work in astronomy, but also the scientific solutions in the nation's needs that is modeled by the National Medal of Science and Technology, which Paul Alvasados won. We are based on unique scientific capabilities and facilities that are larger and at a scale that universities can't operate, that we do, and advanced instrumentation, and can manage these large research teams. So we're particularly well suited to tackle these national challenges as they come up. And finally, the most important part of the laboratory are our diverse group of highly trained, creative, and committed scientists and engineers, and you're hearing from some of them this evening. The fact is the national laboratories were invented to deal with national challenges and national emergencies. In fact, this is from 1939. The photo of the radiation lab staff, the precursor of our laboratory. And that is E.O. Lawrence there at the age of 39, the year he won his Nobel Prize for the cyclotron. Up there is J. Robert Oppenheimer, who was a 38-year-old theoretical physicist before he took on his role in the Manhattan Project. Robert Wilson, who is a 25-year-old graduate student. He built the biggest accelerators in the world at Fermilab, and I was a director at his laboratory. I was the 4th director there, and I'm now the 8th director here at Berkeley. And Luis Alvarez, who developed ground-based aviation control using radar in the war afterwards. Then he built bubble chambers that investigated the nature of matter and won the Nobel Prize for that. This was a remarkable set of scientists who realized that by doing things together in big teams, they could tackle problems nobody else could. And that is our tradition, and that's what we're applying here. Although, as I always point out, this group of scientists at the top here were all white males, and that's not the way our laboratory looks today. So our laboratory, as many of you know, is the main priorities for us are chemistry and material science, computing sciences, fundamental physics, and biological environmental science. But in fact, we also have a central part of our priority now is infrastructure renewal, because we have, in some sense, $1 billion program over the next several years to build the future laboratory, the laboratory that we need in the next 20 years with a series of new building projects. So our COVID-19 research is remarkably broad as the laboratory is. We have X-ray and laser experts who are working with biologists to study the structure of viruses and how they work within droplets, so they can study this in real time and understand how, in fact, they can control the infectiousness. We have people who have built a text mining tool that they use in for other areas of science, which they brought in to synthesize the mountain of scientific literature on COVID-19. It's become a world resource for people working in that area. Our lab supercomputers are being used to model how the coronavirus enters the human cell and helps understand the process and the structure of the spike. They're also being used for epidemiological modeling. And our big facilities, the Advanced Light Source, which is one of the handful of facilities in the country where all biological molecules are analyzed for their structure, has been working on this problem. And in a recent Nature article, scientists showed that antibodies derived from the SARS survivors, the closest precursor of SARS-CoV-2, could potently block entry of SARS-CoV-2 and other closely related coronaviruses into host cells. So that may be an important fundamental discovery for developing therapies. Finally, in one of our newer research projects showing up there, using our people who study air quality in buildings, doing experiments on airborne virus transmission in buildings to study the transport and fate of aerosols. We know that this is far less infectious in situations outside just because of the air movement. And what can we do to control air environments so that we can do better indoors? So for anybody who's interested in what we're doing besides what you hear tonight, there is a nice website where this research is being collected. It's shown here. I invite you to take a look at it. And with that, I'd like to thank you and say that we're happy to, although our laboratory has been taken over by the turkey and the deer mostly over this time with very few people, we're happy to starting to be back to work to do what we do best, which is our world-class research. So with that, I will stop sharing. There we go. Okay, and thank everybody for being with us tonight. And I hope we'll have a chance to do this in the future and soon, actually, in fact, at the lab. Thank you. So with that, I'm going to turn it over to Michael Connolly and to have him go back to going through our research.
Thanks again, Mike. and thanks for this opportunity to speak to you all about some exciting efforts at the Molecular Foundry related to COVID-19. I'm Michael Connolly, and I'm a Principal Scientific Engineering Associate at the Molecular Foundry. The Molecular Foundry is a Department of Energy funded national user facility located at Berkeley Lab that serves more than 1,000 academic, industrial, and government scientists around the world, including researchers from nearly every state in the nation each year. The Foundry is a nanoscale science facility where users come to perform multidisciplinary research utilizing our cutting-edge instrumentation and staff expertise. As I mentioned, at the Molecular Foundry, we study materials at the nanoscale. And if you don't already know, the nanoscale is very, very small. A single sheet of paper is 100,000 nanometers thick, and a single COVID-19 viral particle is about 80 nanometers across. We're talking about really small scales. For some historical context, peptoids were developed in the early 1990s by a Bay Area biotech company called Chiron that had a wealth of new drug targets thanks to a revolution in genetic engineering, but lacked the chemical expertise and large compound collections that were the key tools used by traditional pharmaceutical companies for drug discovery. Peptoid technology, including the chemistry and the automated synthesis methods, became the chemistry engine that they used for drug discovery. You may remember from biology that peptides and the larger proteins are built out of amino acids and have many amazing functions in living organisms, including providing structure, serving as chemical messengers, binding to targets like a virus, or catalyzing chemical reactions. Simply put, peptoids are an artificial biopolymer that mimics these naturally occurring peptides in protein. However, peptoids are more robust, easier, and less expensive to produce. We like to think of peptoids as occupying the space between natural protein and industrial plastics. In 2006, I was one of the early scientists to join the Molecular Foundry and was fortunate to get to bring with me a couple of the peptoid synthesis robots that I'd helped develop at that biotechnology company I told you about earlier. These robots were the beginning of an ever-growing assortment of automated synthesis robots that we make available to our users at the Molecular Foundry that make a wide variety of nanomaterials, including the quantum dot nanocrystals that may be in some of your televisions today. Since then, we've assembled a suite of instrumentation that allows us to rapidly synthesize, purify, and characterize peptoids. This facility is enabling because it allows non-chemists to create designer molecules to drive their science and help to answer questions across many scientific disciplines. Peptoids have provided nanoscience solutions to many challenges our users face, ranging from research into new coatings that allow naval ships to more efficiently travel through water, molecular Velcro for binding bacteria, to a local startup company named Xtherma, who is mimicking antifreeze proteins found in Arctic fish to help to extend the time of donated human organs for life-saving transplants. Today, I wanted to highlight two of our user projects using peptoids as possible solutions to our current COVID crisis. The first project is led by Professor Anneliese Barron from Stanford's Bioengineering Department. She has been working with the Molecular Foundry to develop peptoids that mimic natural antimicrobial peptides. These antimicrobial peptides, also called host defense peptides, are part of the immune response found in multicellular organisms, like the African clawed frog that produces a peptide called meganin. The human immune system also includes similar antimicrobial peptides. These antimicrobial peptides have been demonstrated to kill bacteria, viruses, fungal, and even cancerous cells. Anneliese's group has been designing antimicrobial peptoids and sending them out to a network of collaborators across the country for screening against a large variety of disease targets. This screening by Annalise's collaborators, including Dr. Gil Diamond at the University of Louisville in Kentucky, has identified peptoids that show significant antiviral activity against herpes simplex and hepatitis C virus. Because of this significant antiviral activity, collaborators were interested in screening these antimicrobial peptoids against the novel coronavirus now causing a worldwide pandemic. In late March, during the lab's curtailed operations, I led a small team, including members from Annalise's lab, that rapidly completed the synthesis of these key peptoids so that they could be sent to Dr. Kenneth Stapleford at NYU's Langone Medical School in New York City for testing against the active COVID-19 virus. We've also sent peptoid samples to virologists in New Zealand for testing against a novel strain of COVID-19 they isolated there. We stand ready to scale up any promising leads for additional development. Our second user project is led by Dr. Stanley Chi, also from Stanford. Stanley is harnessing the power of a primitive immune system that bacteria use to fight off viral infections. This primitive bacterial immune system is referred to as Crispr. Stanley's system that he has dubbed Pac-Man for prophylactic CRISPR in human cells uses RNA fragments from the COVID-19 virus that act as homing beacons and direct an enzyme to chop up the viral genome. Stanley has designed 6 of these CRISPR RNA sequences that can target more than 90% of all known coronaviruses. Stanley's system has the potential to be a powerful weapon not only to COVID-19, but also against future emerging strains that have pandemic potential. However, to be developed into a viable therapy, it must be coupled with an effective delivery system to get into human cells. Our lab has developed lipid peptoid hybrids that we call lipitoids. Lipitoids self-assemble with RNA or DNA in solution into nanoparticles. These lipitoid nanoparticles have been demonstrated to very efficiently deliver nucleic acids into mammalian cells, including cell types that are very difficult to transfect. And these are now being studied by clinical scientists for therapeutic uses. The Chi Lab is working to test CRISPR using our lipitoid delivery system. These initial experiments have been quite promising, and the Chi Lab has demonstrated that they can effectively degrade the SARS-CoV-2 sequences and influenza A virus genome in human lung epithelial cells. They are currently working to optimize their protocols and will be sending these formulations on to our collaborators for testing against active COVID-19 virus. In summary, I believe this work at the Molecular Foundry demonstrates the power of our peptoid platform when leveraged with the innovation of our user community to rapidly supply novel nanomaterials for a multitude of applications. I want to acknowledge the hard work of Dr. Tasha Malchanova and Claudine Herlin, who pushed forward during our curtailed operations to complete the synthesis of the key peptoids. Professor Annalise Barron, who in addition to being the driving force behind the antiviral peptoid projects, also serves as our current chair of our Foundry User Advisory Committee. Professor Stanley Chi at Stanford for his Pac-Man CRISPR antiviral therapy project, and Dr. Gil Diamond from the University of Louisville, who tested many of these peptoids and identified antiviral activity, and Dr. Kenneth Stapleford in his lab at NYU Medical School, who is currently testing the peptoids against active virus under extremely challenging environment in New York City. And with that, I'll hand off to Deepti Tanjore, who will tell us about work at the ABPDU.
Thank you, Michael. So, hi everyone, my name is Deepti. I'm the director of ABPDU. In the background here, you're seeing a goat. At the lab, we rent goats to eat the dry grass off of the land. So we can avoid wildfires. I find that very fascinating. Hopefully that's not too distracting to you. Today, I'm not going to talk about goats, but I'm going to talk about biology and how biology can be leveraged to be able to solve some manufacturing related problems within COVID-19. So as I mentioned, I'm the director of ABPDU. ABPDU stands for Advanced Biofuels and Bioproducts Process Development Unit. We are a scale-up facility that is funded by Department of Energy Bioenergy Technologies Office. We are available to help industries and academic entities to solve scale-up problems, to identify and solve scale-up problems. The reason this is necessary is because many lab-scale technologies are developed in multi-well plates in very small volumes. So the experience that a culture has in those well plates is very different from that in a bioreactor, where there's a lot more turbulence and the materials are much more viscous in nature. So we have to re-optimize many of the technologies that are developed at the lab scale in liter scale reactors and then scale them up further to hundreds of liters. We've been doing this kind of work for over 8 years now. We've collaborated with over 50 companies and several academic institutes as well. We typically work in the biofuels and bioproducts area, but in the past couple of months, we have been focusing our efforts, our expertise and equipment to help solve the COVID-19 problem. So I'm going to give 2 examples of such projects. The first one is with a company called SwiftScale Bio. They are using cell-free manufacturing platform to produce antibodies for COVID-19. And another project is within our division to produce probiotic vaccines that can be delivered by probiotic bacteria. So in the first project, before I go ahead with explaining SwiftScale, I want to briefly give you an idea of what it takes to manufacture either an antibody or a vaccine. For the first, before we even scale up, we have to first develop a strain that can produce these molecules. And once we produce that strain, then we can scale them up to hundreds of liters, produce these happy bugs with our molecules of interest. These bugs are happy because they have a lot of salt in them, and the culture has a lot of water in it. We give all the ingredients that can keep the bugs growing and producing the molecule of our interest. But when we need to extract the molecule, the first thing we have to do is centrifugation to remove the water from the cells, but we still have the salts left with it. So we wash off the salts with extra water and buffers and filter out the bugs that the bacteria that is producing the molecule of our interest. But the molecule is inside the bacteria, so we have to break the cells open and then produce that molecule or recover that molecule via purification, chromatography and purification, and then we have our vaccine or antibody. So it's a scale-up manufacturing is a very involved process. So one of the problems in this is around not being able to generate the amount of molecule that we want to produce in one go, because cells typically do not produce a large volume of the product that we need. So SwiftScale Bio has been looking at using cell-free manufacturing, which means that they grow the cells not for the molecules, but the enzymes that make the molecules. And then they harvest the cells for the enzymes and do the enzymatic reaction outside the cell. So that's why it's cell-free. This way we can produce a lot more amount of the molecule than the cells can produce inside themselves. SwiftScale has been around only for about a year plus now. They're a very new company because cell-free manufacturing in itself is a very novel technology. The ABPDU has experience in this space, so they came to partner with us and we helped scale up their technology. A couple of weeks ago, we scaled up from a milliliter we produced inoculum to inoculate the next level to 10 liters. So we call them seed 1 and seed 2, and then eventually we went up to 300 liters scale up and then provided the cell pellet for them to harvest the enzymes from and do further studies on antibody production. So the next project I want to talk about is around the probiotic bacteria, as I mentioned before. This work is being conducted or being led by Deepika Avasthi, who's a project scientist at Joint Bioenergy Institute. She is looking at trying to clone probiotic bacteria with the vaccine that is needed to be delivered in a human body. The idea is that we take the proteins from coronavirus, so it's not the entire virus, it's just one of the proteins, and be able to clone them and express them in probiotic bacteria, so we can eat them in food or other ways. In that way, we don't have to break the cells open, so that reduces half the manufacturing line needed for the vaccine production. And also, the consumables needed to provide an intramuscular vaccine, like syringes and also healthcare worker resources, can all be reduced. And needless to say, the experience of taking the vaccine can also be much, much better. So this approach is, again, to reduce the costs associated with manufacturing and scaling really fast. The ABPDU and the Biological Systems Engineering Division has been involved in several other projects that are ongoing. One such project is with a company called Crispr Bio that is looking at generating kits that can rapidly detect SARS-CoV-2. They were recently even highlighted in a New York Times article for using CRISPR-based enzymes to develop better assays. We've collaborated with Argonne National Lab to apply to some funding via the RAPID Institute. It's an institute that focuses on process intensification. We are looking at rapid antibody purification work through them with funding from NIST, National Institute of Standards and Technology. We're also collaborating with the University of California to scale up antivirals production. And then Sandia National Laboratory and Lawrence Berkeley National Laboratory are collaborating in being able to develop and get another assay around digital microfluidics, something that Rebecca has mentioned earlier in the presentations. So with that, I'd like to thank the ABPDU team for all the effort they put into these projects. And I also want to turn things back to Jennifer.
Thank you so much, Deepti, for your presentation. And thanks also to Michael, Rebecca, and Mike for their presentations. Now we'll begin our Q&A session. And as a reminder, please submit any questions you have using the chat function at the bottom of your Zoom window. Just click on the chat icon, type your question, and press enter to send it. Now we've got a couple good questions, and I think I'd like to start with this one. I think it might be interesting to hear from all of our panelists, but let's start with Rebecca. How quickly were you able to pivot from your current research to working on COVID-19 research?
Well, I think I started being involved in daily meetings the day we were sent home from the lab. So that would have been, I don't know, March, something, the first day this all started. And I think in terms of understanding the framework and moving from one type of emergency to the other, but still coordinating with the same agencies and the same stakeholders, it's not much of a pivot. In terms of the technical work, this has involved quite a lot of people. And so it took a few weeks to put in place. It took a few weeks to receive funding to actually start doing the work. But I think it was very quick considering the situation. So I would say a few weeks.
Thanks, Rebecca. Michael or Deepti, would you like to take a stab at that question?
I'll go. I think, you know, we knew in mid-March that, well, our user, Annalise Barron, was working on these antimicrobial peptides, and we knew there was interest from collaborators then. We started synthesis, and then when the lab went into curtailed operations, it took us about 2 weeks before we were sure that we could do the work safely and quickly. And we got back in at the end of March and spent about a week doing that initial synthesis. So we were able to turn around pretty quick on that. And then through Annalise, we were actually introduced to the Stanley Cheese Lab, who had been working with the CRISPR system for COVID. And actually, he had been working on the flu virus, and he turned around his work to focus on COVID-19. And then we made that connection, and we actually had a stockpile of the Lipotoid available. So it was mainly a matter of getting to our -80 freezer and getting a sample sent down to Stanford for him to begin that work.
For ABPDU, we were talking with companies even before the shutdown because they were looking to generate solutions even where this problem existed in other parts of the world. So we started talking to them and started the contracting process at that time, but making sure that we execute it safely and also making sure that we're doing it right way did take a little bit of effort from our end. But it was surprising how much we could use our experience and expertise that we already have that came in handy in being able to cater to these projects, which are not necessarily producing molecules similar to biofuels or biochemicals. So I'd say a few weeks, but yeah, it was faster than I expected it to be.
Hello, and this is Dan with the Labs Communications Group, and I'm helping to relay some of the questions coming in to our panelists. And this next one goes back to Michael at the Foundry. The question is, are the lipotoid precursors or the self-assembled lipotoids able to pass through human skin? And do you have to consider potential toxicity when you work with these?
Okay, so as far as delivery through the skin, we haven't explicitly studied that, but we have studied against a large panel of different types of cell lines through the years. And when we do that, we were always optimizing for delivery versus toxicity. So we screened to find the best peptoid nanoparticle formulations that gave us the best delivery of the cargo with the least toxicity to the cells. Now, more recently, there have been studies in animal models looking at damages to, say, vocal cords and such, and there the peptoids have been found to be non-toxic. For COVID sorts of delivery of Stanley's Crispr system, they were thinking more along the lines of delivery via an inhaled solution rather than through the skin.
Thanks, Michael, for that answer. We've got another question coming in, and this one is for Deepti. Deepti, what are some of the most challenging obstacles to overcome in scaling up bioproducts like coronavirus antivirals? And how quickly can products be commercialized after scale-up challenges have been addressed by the ABPDU?
Yeah, so the scale-up process, for example, antibodies or vaccine, it needs some research work, right? So we have to do some process development at hundreds of mil scale first for several weeks, and then we would scale it to hundreds of liters. Usually for molecules that are in the pharma side of the industry, hundreds of liters or thousands of liters are sufficient, 1,000 or 10,000 at most. Actually, 10,000 is also pretty high. So scale up is generally a little bit faster than the biofuels and bioproducts industry. But the purification part is where the challenges come in, because if you're putting a biofuel molecule in an engine and burning it, purification is not as much of an issue as you're putting a molecule in someone's body. So we have to be very thorough with our purification process development. And also manufacturing has to be in a CGMP facility. ABPDU is not a CGMP facility, but we can do all the work to develop the process and then hand it over to a CGMP facility for manufacturing. And that's the kind of work we're doing right now.
Thank you. And here's another question that has come in. And this one is for Rebecca. So what are some of the challenges to optimizing or improving the ability at the R&D level for high throughput workflow for COVID-19 testing? What are some of the big challenges there?
I think the main challenge has been to make sure that we can operate the instrumentation safely. And that, for example, while you can do manual type of experiments in a biosafety cabinet and make sure everything is controlled, while you're operating larger scale instruments that are going through an automated workflow, you may be creating aerosols and you have to control this, especially if we're talking about a virus that is so infectious. And so I want to say in this case, the main challenge has been to repurpose some of our instrumentation so that it can be operated safely. That would be it.
Thanks, Rebecca, for that answer. I've got a question coming in, and I think, Mike, you might be the best person to answer this. In light of the COVID-19 crisis, is there an accelerated process for researchers from academia or industry who want to access, maybe the Foundry or the ABPDU or some of the other facilities at the lab?
Yeah, so as you heard, the pivot of this research happened actually so fast. Usually getting new research funding and getting something started is something that takes a year, and this is done in weeks. That could be done so quickly because it was done at the laboratories with all the ways of getting things approved and funded at the laboratories quickly. The Department of Energy programs are looking at how do they extend this to a larger group with time. And that's something they will be doing that. We're not in control of that. They will do it from their offices, but they hope to actually broaden this. The thing that's important for us is when we can get actually visiting users at our user facilities. Right now, we only have a few people who are on our site with special cases where our employees are doing research sort of with university researchers which are working remotely. We're working to make our user facilities available over the summer so that we can get to the point that we can actually have normal users come in. So as soon as they're able to travel, we can actually have them come in.
Thank you very much. And this next question is kind of an open-ended question. Turning on making sure my video is on as well. The question is, when might we be able to expect kind of widespread vaccination to become available? And that's, if any panelist wants to take this on, that could be just an interesting question based on the state of the science we have now and the rate of development.
So who wants to take that one on? This is the most difficult question, I think, in the whole scientific worldwide community right now. And it just, on the one hand, there's an enormous pressure on getting things done faster than they've ever been done to test vaccines. And the fact is that actually no vaccine's been done in less than about 1.5 years in history. And usually it takes longer. So I think that's as much as anyone can say right now. What you read in the news about this is as much as anybody knows at this point, which is a lot of testing is being done, and they're trying to accelerate the process, but it usually is a process that is very difficult and takes something like 1 year or more to do.
Thanks, Mike. Do any of the other panelists want to take a stab at that question?
I will say that, we see from users that are trying to push forward with new vaccine technologies, and I'm sure people may have heard about, the mRNA viruses and those sorts of things. And the lipitoids that I told you about are those lipitoid nanoparticles are very similar to the particle delivery systems that some of those the mRNA vaccine companies are using for delivery. So there's a lot of new technology that's being developed that will hopefully short circuit some of those timelines, but it's all new science and we're just doing the best we can and working hard every day.
I can also add manufacturing it and providing it to 330 million people in this country and all over the world can be a very demanding, a very, very difficult task. So the probiotic vaccine project that I mentioned earlier is one such attempt at solving such problems by being able to deliver vaccines through food or innovative ways where we don't have to produce the syringes, we don't need to go to the hospitals and maybe avoid further infections by going to hospitals. So yeah, there are many ways, many angles at which people are coming at it, and hopefully we'll have a solution soon.
Thanks, Deepti and all for answering that question. We've got another question coming in. This one's for Michael. And the question is, for the technology that you discussed, what kind of time frame would be needed to fully test them for effectiveness in the real world?
So, for the peptoid, antimicrobials, those are still really early in the development pipeline. And, the CRISPR being used as a therapeutic is a completely novel, therapeutic. So those would have to go through all of the phases of FDA testing, and it's usually a very arduous and long process. That said, a lot of the, materials that are available today for therapeutics and vaccines that are, towards the front end of the line on that development cycle were initially developed or started against other diseases, be it SARS or MERS or that sort of thing. So, the things we're developing today may not actually end up being effective or in use against COVID-19, but they might be available for COVID-20 or 21 or whatever the next, pandemic is that hits us.
Thank you. And this is a question for Mike. How is the COVID-related research happening at the lab right now prioritized among all of the other kinds of research that the lab undertakes?
Yeah, so what happened in the standby period we had between March 17th and last week, the COVID research was given basically very high priority. That is, it was almost all the research that was actually going on our site. Everybody else was working remotely. But starting this week, we now are having a small amount of research in every part of the laboratory. And so now the other mission critical research is being leavened in. So none of the COVID research was stopped. It's still going on. But now we have a balance of both our, if you will, conventional, usual research program along with the COVID research. And over the course of the summer, we'll bring more and more of that in. So the COVID research has not stopped any of the other research. What stopped the other research is just the access to the site. And as soon as we actually get full access to the site, we'll be able to get full restart again.
Thanks, Mike. I've just noticed that hour went by quite quickly. It's 7:30, which brings us to the end of the presentation. Before we close, I'd like to thank Mike, Rebecca, Michael, and Deepti for their presentations. And I'd also like to thank the audience for tuning in and for your interest in research that's happening at Berkeley Lab. If you'd like to stay up to date on the lab's research, you can visit us at www.lbl.gov, or you can follow us on social media. Our handle is @Berkeley Lab. Thanks again, and have a good evening, everyone.